Welcome to the Data Science Institute's virtual seminar series. My name is Dr. Sarah Mackey from Lawrence Livermore National Laboratory. Here at Lawrence Livermore, data science has become an essential discipline in many of our key program areas. LLNL is home to many challenging datasets, as well as home to some of the world's most advanced supercomputers. Our data science staff work in a variety of areas, including machine learning, artificial intelligence, big data analytics, statistical inference, predictive modeling, and uncertainty quantification. The Data Science Institute acts as a central hub for our lab's data science activities. We host events like this seminar series in order to introduce new ideas and potential collaborations to our laboratory staff. We invite speakers from outside the laboratory, from the Bay Area and beyond, to share their innovative approaches with our data science community. We're pleased now to include a wider audience in our seminar series through these recordings. You can read about past speakers at our website at data-science.llnl.gov, or you can email us at datascience@llnl.gov. Thank you and enjoy the seminar series. Today we have Professor Nitin. He is joining us from UC Davis. He is a faculty member there in the Department of Food Science and Technology and Biological and Agricultural Engineering. His research is at the interface of biomaterial science, biosensors, mathematical modeling, and data analytics. His research aims to enhance the quality and safety and sustainability of food systems. He is an author of 145 publications, a holder of 10 patents, and the co-founder of 2 early-stage companies. So Nitin, thank you for joining us today, and go ahead and begin when you're ready.
Yeah, thank you. Thank you, Sarah, for the kind introduction and for the kind invitation actually to present here. So I'll be presenting to you some of the work that we have been doing with AI-enabled innovations in validation of sanitation and detection of pathogens. Before I start my presentation, I'd like to acknowledge the students and the postdocs who actually did majority of the work, and as well as collaborators from UC Davis, as well as North Carolina State University, who were part of this project's different sections of my presentation. Next slide, please. Sarah, next slide, please. Yeah, thank you. So before I start, I'd like to introduce this recently developed NSF USDA funded AIFS Institute, which is focused on AI Institute for Next Generation of Food Systems. This is centered at Davis, but has other collaborators in this part of this institute. The goal of this institute is to develop AI technologies to help produce the next generation of food systems, which are sustainable, nutritious, as well as use fewer resources through AI enabling technologies. So some of the projects that I'm going to share today are motivated by our research as a part of this institute. Some of them started before this institute, but were part of founding the institute overall. Next slide, please. Now, when we think of agriculture has gone through many revolutions over the period of time, dating back even pre-industrialization. So there was an industrial revolution that brought tools, mechanization that helped enable agriculture to scale from small farms to large farms. And then we had extensive development of agrochemicals, the nutrients and the monoculture crops, which allowed us to develop optimal crops for production and growth of agriculture as a part of it. And as we think about 21st century, we're thinking about agriculture as a part of delivering precision nutrition to us, helping us select crop varieties that produce the right nutrients, the right crop yield, minimal utilization of resources. So again, these are part of the journey that agriculture has made along with our journey on this planet. Now, there have been significant enablers in this journey. These are the sensors and devices, some of the AI and data analytics part, as well as the global distribution supply chain that has enabled the growth of agriculture and the food systems, as well as consumer demand for more personalization, as well as awareness of health and food relationships. At the same time, there are concerns with global safety and sustainability that also enables some of the innovations in this space. While there are these enablers, there are also barriers in this. One of the key barriers that we found in developing this center as well as in our research is that there is a big gap between agriculture practice and the data analytics or the AI systems. So how do we bring these two together so that we can reduce the innovation gap from the technology side as well as from the agriculture side, as well as the education and outreach to train the next generation of workforce that can reduce these barriers further. Next slide, please. So the AI Institute in Food Systems based in Davis is looking at a holistic approach to food systems. We have specific research thrust and clusters. These include molecular breeding, ag production, food processing and distribution, nutrition, economics and ethics, as well as social sciences. So these are the specific clusters. And in all these clusters, AI and the technology are the enablers. So they enable the innovation in these respective areas. Next slide, please. As I mentioned, this is a multi-institutional center funded by NSF and USDA. The organizations involved include 6 organizations: UC Davis, Cornell, UIUC, UC Berkeley, USDA ARS, and UC ANR. And it involves a significant number of faculties, about 46 faculties and 7 integrated teams working together to address these goals of the institution. Next slide, please. So I co-lead the food processing and distribution cluster, and this cluster is focused on 3 core goals. These goals include the food safety and traceability, food loss and spoilage, and the process innovation and optimization. And the team represents diversity of researchers from Cornell, UC Davis, UC Berkeley, UIUC, and USDA. Next slide, please. So the food processing and distribution cluster, some of the key challenges and goals, these goals are something that we all connect to. I think we all have heard the news about food safety, the risks that are associated with the supply chain. And these risks actually involve significant interactions with the environment, with the people, with the machines that we use to move the food from farm to the fork. Similarly, along this supply chain from the harvest to the consumer, there are significant chances of food loss and spoilage. Some of these are related to physical processes, our ability to maintain temperature, for example, or moisture in some cases, microbes, as well as the supply chain and our behavior as consumers. The third challenge in the food processing cluster that we often hear about is that, food manufacturing industry is an intensive operation that uses a significant amount of water, energy, and chemicals in some cases, especially when it comes to sanitation. So how do we develop processes that reduce the use of these resources to make the industry more sustainable and more resilient? Next slide, please. So beginning from this slide, I'll talk about some of the things that we are doing in my lab. So I'll shift focus from the center to more focused on some of the things that we're working in my lab space here. So when we think about food supply chain, we think about pre-harvest, harvesting, processing, and retail environment. And often these microbes, which are pathogens or spoilage microbes, which are not truly part of food system, can hitchhike a ride. So if you think about, pathogens often are hitchhikers in this process. So they enter at certain points in the supply chain and can continue distributing them. And once they enter the supply chain, they can propagate, cross-contaminate, and eventually lead to outbreaks that lead to hospitalization and in some cases, deaths of individuals. Next slide, please. So I guess my slides got a little bit messed up in terms of the view here, but I'll illustrate the key point here regardless of it. So when you think about the outbreaks, now fresh the plant-based food has a very little caloric intake associated with it. Processed foods, animal food, they constitute more than 80% of the calorie intake that we take. About 15 to 16% of our calories come from plant-based foods. These are fruits and vegetables, seeds, nuts, included those. And if you look at it, this particular category of fresh produce accounts for significant numbers of cases. These are like close to 60% of Listeria cases, Salmonella cases, and significant number of viral outbreaks during the last decade. So there must be some reason that this category of fresh produce leads to significant number of outbreaks in our society. Next slide, please. So one of the reasons we believe that there are high risk in this category is the risk of cross-contamination. Now, just to illustrate the point, the cross-contamination is a process by which microbes enter a food supply chain or enter at some point a product, and then from that contaminated product, we cross-contaminate other non-contaminated products. And this has to happen for a nationwide outbreak. Without a cross-contamination event, it's very difficult to actually have a nationwide outbreak. If there was only one apple or one spinach leaf that was contaminated and it did not cross-contaminate the others, we would never have a nationwide outbreak. So the only reason that we hear about nationwide outbreak is that there is a cross-contamination event that takes place along the supply chain, sometimes along the processing supply chain, sometimes along the distribution supply chain. Now, while we take these products, this product comes into contact with water. Water often becomes a source of cross-contamination. Often there are organic matter when this get harvested, especially with mechanical harvester. We put a lot of organic matter with it, produce residues, dirt. And these all residues and dirt that comes with the product, we try to wash it away, but carries a certain risk of cross-contamination. And as a result, this process is also highly variable. Now, this introduces a significant challenge because when you have highly variable processes, how do you develop process validation tools, which I'll talk about, or process verification tool, which I'll talk about next in my presentation. Next slide, please. Now, when we think about food safety, there are two major approaches that the industry uses. The first, I'll call it a control approach, where we try to prevent microbes from entering the food supply chain. And we also develop processes that try to eliminate microbial contamination, like pasteurization. When you buy milk, your milk is pasteurized. And one of the key steps is a thermal treatment process that actually reduces the microbial contamination of the milk and improves the shelf life as well as reducing the food safety risks with it. The other approach is monitoring. We do the environmental monitoring for pathogens. We also do testing of food and inputs that form part of our food system. Now, I'll give you examples of some of the research that we have been doing, at least in my lab, focusing on developing controlling processes, as well as process validation for those processes, as well as the monitoring of these pathogens and using AI-enabled solutions to detect pathogens in food supply systems. Next slide, please. So when we think about controlling cross-contamination with optimized sanitation processes, the need here is that when we introduce, when we have a process in industry, it needs to be verified and validated. And especially when the conditions are highly variable, this needs to be done repeatedly and done on a routine basis. So for example, in a sanitation of wash water, we try to introduce chlorine in it, and that is how your triple batch salad is prepared, to make sure that there is adequate amount of chlorine available to inactivate the microbes in a system. Similarly, on a food contact surface, we use sanitizers and mechanical treatments to make sure that we get sanitation of it. Now, when we think about process verification, our goal is to predict how much bacterial reduction do we get for a given treatment time and concentration. It's a very simple task, but it becomes challenging when your process conditions are highly variable. And also when there are spatial variations along the process, like where do you sample in a process condition? Do you sample at where you inject the chlorine in the system, or where you think there may be less chlorine in the system? So all those conditions introduce significant challenge. Or do you evaluate this as a cumulative treatment? So those are the kind of questions that we try to ask, and how AI may enable us to address some of the needs there. Next slide, please. As you heard in the introduction, one of the areas that my lab work is in biomaterials. So we went to this problem to the industry, and they told us that the challenges that they have for process verification and validation is that their raw material is very different, and it also has very different levels of contaminant every time you do it. And if you look at chlorine that they inject, there are significant number of studies that shows that when you try to wash your produce, there is significant variation in chlorine, both spatially and temporally in the system. And these happen because of all the variation that I said earlier. So the challenges the industry has is that they could take the water sample and try to culture it, but the cost of doing that is very high. If they send samples for PCR, that also costs them significant resources, because you have to do multiple tests multiple times in a day. That becomes a significant cost to them. And some of the tests can take a long time. They can also look at water chemistry, but water chemistry question is, where do you measure water chemistry? If you measure very close to where you inject chlorine, yes, you can measure it, but if you spatial and temporal variation is difficult to capture. And often some of these water chemistry measurements may not represent what happens to the sanitizers when they interact with microbes. So our solutions coming from a biomaterial side was that, can we develop non-living edible surrogates? So these materials are made with food. They're non-living because food industry does not want to introduce anything living in the processing plant. And so we developed the materials, and with the AI aid, we developed detection algorithm using applied machine learning approaches that enable us to predict the efficacy of the treatment. Next slide, please. So our edible surrogates are such that they could be immobilized on the produce itself, both in form of easy readout stickers, or they can be immobilized as a part of the product. And we can tune them so that they have that they can stick to those materials. We acquire the data using spectroscopy from those sensors or edible surrogates that we prepare, and then we use machine learning or statistical prediction algorithms for process validation and verification. So that's the innovation that we enabled in this space. And next slide, please. So just to summarize, these edible surrogates for process verification and validation are made with edible material. They're non-living. As I mentioned, industry is very particular about introducing living species into their plants. They have high affinity to bind fresh produce or other surfaces, such as food contact surfaces. In the food industry, cost is a big factor, so they have to be cost effective, and they provide rapid and quantitative measurements, which is culture free. And they predict the sanitizer concentration and microbial inactivation levels. Now, what we are working on is to improve the robustness of our algorithm for prediction efficacy, as well as developing portable and bench scale systems that they can be deployed in the field. Next slide, please. So I'll share some of the results on this strategy that we're working on. So our ideal is that we have wash water in which we add sanitizer. And as we add this sanitizer results in inactivation of microbes. So similarly, when our edible surrogates interact with this sanitizer, we get an FTIR spectrum that provides us a measure of the output from those. We try to do the correlation between the prediction of the microbial inactivation and the spectral response from the edible surrogates, using some of the well-established machine learning approaches. And we try to classify both the dosimetry and the level of inactivation based on this predictive model. Next slide, please. And now, before we started doing this, we were actually working with some standard classification models, such as PCA and other models. One of the drawbacks we found was that these clustering approaches were not able to classify them into categories, especially when we took data across multiple days or multiple sample types. And clearly, the introduction of noise in the data made some of these conventional approaches not robust enough to predict either the dosage or the level of inactivation. Next slide, please. So I'm showing you the first of the results here, where we are using chlorine as a sanitizer, and we are using a DNA-based edible surrogate. Using this surrogate, we can predict the level of sanitizer concentration, as well as we can predict and we have fairly high true positive rates and very small rates of false positive across these categories. So we can predict when our surrogates are being exposed to 15 ppm of level of chlorine versus 2 ppm level of chlorine, using the classification model that is built based on a light GPM model in this case. Next slide, please. Now, one of the goals is in the industry has been that, okay, we can know the sanitizer concentration, but can you actually tell us without culturing how much E. coli will get inactivated in this case? And this is important because by law, there is a requirement that this water that is used for washing should have no residual E. coli in the system. So it clearly has to be zero. And it's often a problem because if you detect E. coli in your system, then that means that there was a chance of cross-contamination and there may be other pathogens present also. So in this case, we are able to predict, so clearly getting 4 or 5 log inactivation is the desired goal. Often we get somewhere between 2 to 3 log inactivation in these systems. So we can clearly present both the control samples, 2 log inactivation and the 4 log inactivation with very high accuracy, again, using the confusion metrics and the ROC curves clearly highlight that our accuracy in predicting the microbial inactivation based on spectral response is very high. Next slide, please. Now, what I've told you so far has been using a chemistry which is well defined, is single chemical chemistry, which is NaOCl, which is sodium hypochlorite, we call known as bleach also. But what happens when you try to take the same approach and increase the complexity of the problem? Because I think that's where the value of AI even becomes more clear. So in this case, we are looking at non-thermal atmospheric plasma technology. As some of you may be familiar with it, this is the fourth state of matter. We have solid, liquid, and gases, and the plasma is the fourth state of matter. This is a multi-species system now. It's a highly nonlinear system. It produces multiple reactive species, nitrites, nitrates, peroxides. And we are working with Drexel, NC State, and Rutgers actually to scale up this system and for deployment in the industry. This is a 100-liter unit as shown here on your left here. So one of our goal was that can we validate and verify the dosimetry with this system? And this is critical because if this system is to get approval by FDA for food processing operation, it needs to have a dosimetry. Now, when you think of thermal processing or high pressure processing, it's very easy to get dosimetry because you know the temperature time relationship or the pressure time relationship. But when you have a chemical system which is highly nonlinear, how do you describe the level of active chemicals and their interactions with the pathogens or their surrogates in this case? So that's the challenge that we're trying to address with this problem at this stage. Next slide, please. So our experimental approach is similar. We have a biofilms on a stainless steel surface that we grow, and we have our surrogate, which is again based on a DNA biopolymer mixture. We subject them to both to plasma treatment. We measure the level of inactivation of biofilm and the DNA FTIR spectra. We use FTIR because it's a low-cost spectral measurement system compared to some of the other approaches. And again, do the classification based on the correlation between these using the classification method of light GPM in this case also. Next slide, please. So we were quite pleased by the results that actually we can predict the dosage of plasma. Now, in this case, we change the time of exposure. So we can tell that this sample has been exposed to plasma for 2 minutes, 3 minutes, the different classes that we have in this case with very high accuracy. So we can predict how much is the plasma activated water dosage based on the spectral response of our surrogates and the machine learning classification model. And next slide, please. So the other next, as I illustrated in the previous example, the next task is, can we actually predict the inactivation of bacteria? Now, this is the planktonic bacteria, which means the bacteria is suspended in water. And we are trying to predict the inactivation of this bacteria. Again, the class 0, class 1 indicating 1 log inactivation, and class 2 is indicating 5 log inactivation. So we're fairly accurate. There are still some challenges in predicting class 2. I think one of the challenge with this is that we have asymmetric data set. As you can see, class 2 has a large number of data sets in their category as compared to class 1 and class 0. And I think that does cause some issues with the classification model in this case. But overall, we are quite reasonably in terms of predicting the microbial inactivation. Next slide, please. And here we are trying to predict the biofilm inactivation based on the spectral response. And again, in this case, we are able to predict 2 log level of inactivation versus 4 and 5 log levels of inactivation of the biofilm based on the spectral response of the DNA surrogate. And both the confusion matrix and ROC curves support that result overall. Next slide, please. So I'll be glad to take any questions at this point, but I can also take questions later. I just wanted to give you guys a moment to ask if there are any clarification or questions at this stage that you may have before I start with the next topic. If not, then I'll continue along the path and we'll be glad to take questions later. Now, as we know, we have been talking about control approaches. We are using a processing technologies to inactivate microbes and make predictions based on the dosage level or the inactivation level. The other way to improve food safety is through bacterial detection technologies, especially the detection technologies that can be deployed in farms or in the processing environments. It is challenging because currently the bacterial detection technologies rely on taking a sample, sending the sample to a specialized lab, culturing these microbes on plates, and it's a time-consuming process. There are some molecular processes such as RT-PCR and other methods, but these are often used either in conjunction with these traditional methods once you get a positive to confirm, or they require significant resources in terms of trained personnel, and often they can't detect live and dead microbes because the presence of antigen or the DNA can still be present if the process has inactivated the microbes. So the goal was to develop a simple and low-cost detection approach, which would only detect viable bacteria in complex food samples. So the next 2 examples that I'm going to share this common goal in this case. Next slide, please. Now, before we started this project in this direction, we were looking for technologies that people have adapted and deployed them in the field actually successfully. So one of the technology that we came across was using fluorescence spectroscopy. This technique has been deployed in the developing world environment as well as in lab-based environments here, where the idea is that there are certain molecular species in water, such as tryptophan, like fluorescence, which indicate the presence of humic substances or bacteria, and can be an indicator of contamination. Now, this approach does work very well when you have a high load of organic content. This approach is not very efficient for detecting low levels of microbes, and clearly not effective for detecting any specific types of microbes. So it doesn't distinguish between E. coli or Salmonella or Listeria, for that matter of fact. But this approach is one of those approaches that has been deployed successfully for online monitoring. So inspired by this approach, we looked into using this approach with some molecular specific approaches to detect bacteria Next slide, please. So the approach that we use is based on using a combination of a bacteriophage and a target bacteria. So for example, bacteriophage is a virus that infects a target bacteria, and after it infects the target bacteria, it produces changes in it. Some of those changes are biochemical changes, and our hypothesis was that these biochemical changes would be specific enough to be distinguished in high organic content background, such as spinach leaves, to distinguish the presence of a particular bacteria. So to do that, we do a simple lab-based preparation that can be done online also. And then we record the fluorescence spectral signature and use machine learning to look at this fluorescence EEM excitation emission metrics to identify whether we have a clean produce or we have a potential risk of contamination. Next slide, please. So our workflow in this case is that we collect the EEM data, and then we convert that into from a 2D matrix, convert the 3D to 2D data set. We normalize the data using Raman scattering from water, and that serves as suppressing the background in this case. And then we do 5-fold cross-validation and evaluate various well-established algorithms in this case to look at their ability to predict the presence of microbes in this system. Next slide, please. So coming to the overall approach, we take the samples with the food materials involved in it, add the bacteriophages to it, collect the spectral response, which is an EEM response. We have a training set and a test set. The first question we asked was that, can this approach actually allow us to detect the classification of different levels of concentration of bacteria? And using this approach, we could detect as low as 10 CFU per ml of my bacteria in this case. We also were selective that if we have non-target bacteria, we do not detect them in this approach, so they do not interfere with our classification model in this case. Next slide, please. So our next thing was that if we have a well-trained model, can we actually use it to test in a realistic food system? And we use a spinach homogenate, which is basically a slurry of a smoothie of a spinach, if you want to consider it. And we collect the spectra, both E. coli with its bacteriophage, E. coli, and the phage by itself. And now, if you notice that the spectral signature has significantly changed due to the presence of spinach in it. Spinach has a huge background in fluorescence domain because it has many chromophores in it as a part of its metrics. Next slide, please. Again, using the same model that we had developed in the earlier stages, we are able to detect both 10 and 10 to power 2 CFU per ml of the contaminating phages, contaminating bacteria with high accuracy, relatively high accuracy, using different models. And they are all within the same range of classification that we can obtain, indicating the presence of bacteria in a spinach homogenate. Next slide, please. So far, I have shown you guys some examples of how we are using spectroscopy and some of the applied ML approaches, which are well established in the field, to look at the data for analysis of food safety and could be applied for food quality and nutritional properties also. And we have shown some predictive relationship between measurements and properties of food systems. One of the challenges is that labeling samples is expensive and time consuming. In some cases, we need to collect more than 100 spectral data sets to make a model. So as a part of the AIFS Institute, we have an active collaboration with Professor Xin Liu in computer science at UC Davis, and she's leading the effort to the data efficiency part of it. So how can we use data efficiency methods to improve the classification and prediction methods without burdening the experimental part of it? So how can we use sparse data or low data or small data, if you want to call it, to build models? Next slide, please. So in this case, we are using an active learning approach, which is also known as query-based learning or optimal experimental design, looking at the utility measure of the unlabeled samples for the currently trained ML model. And using this algorithm, we are able to improve upon the predicted outcome with significantly less number of data that I'll share later in my next slide. Next slide, please. And complementary to it, we're also looking at semi-supervised learning, where we have pseudo-labeled samples, and then we use those unlabeled samples as pseudo-labeled samples to fine-train the ML model. And by using this approach, we are also able to complement the active learning approach in this case to look at the most confident sample that can, according to our model that we have developed in this case. Next slide, please. And our results show that the plasma dosage study that I showed you earlier, using the hybrid approach, we are able to reduce about 50% need for labeled samples and to achieve similar level of accuracy as we needed for earlier set of samples. As I said, number of labeled samples for at least 100 spectral responses were measured. So with the hybrid approach for the plasma dosage classification problem, we can get to less than 50 samples or 45 samples per se. Similarly, for the food pathogen detection problem, using active learning approach also reduced about 25% of the samples. One thing that we observed that is there's no single approach that answers for all data sets, and it depends on the type of data and the application that we could choose. So in some cases, semi-supervised learning approaches were better. In other cases, the other active learning approaches were better. So it's not a universal approach that answers all the questions in this case. Next slide, please. So that I think this slide actually relates back to what I said earlier. So we see significant reduction in the number of samples needed, but different problems benefit from different approaches in this case. Next slide, please. So far we have been talking about the spectral data sets, and one of the other interests that we have in my lab group is to look at imaging-based detection of microbes. So can we actually use imaging as a tool to improve both the specificity of detection and reduce the resources needed, as well as improve the time of detection? And again, this work is in collaboration with Mason Earls at UC Davis, working in this domain. Next slide, please. So again, in this case, we are again using our bacteriophage. It's the virus. As I mentioned, that when this virus infects the bacteria, it causes a lysis. These are lytic phages. So unfortunately, my slide got a bit disrupted there. But if you were to believe me, what you see is a fine-looking E. coli bacteria here, and here you see a smudge of bacteria as shown on the slide here. And this is a result of lysis of the bacteria. So it's a very simple approach that if you take a food sample, incubate for 3 to 4 hours, add the bacteriophage, and if you can have an automatic scanner, you can detect these lysed objects. And so our first goal was, can we develop an AI approach to actually automate this detection method? Next slide, please. And so our goal was to use an AI approach to build a model that can allow us to detect these differences and make predictions of both the count of the bacteria and also the presence of the bacteria indicated by bounding boxes here. And we're using faster RCNN model in this case. And we have compared that to regression and other models, but the faster RCNN model outperforms some of those significantly. So I'll focus on the RCNN model in this case. Next slide, please. So to develop this AI-enabled approach, we took a monoculture, which is E. coli at different concentration. We also took control because we expected that some of our samples that we're going to collect later on may have other microbes in them. Now, these are lab-controlled systems. We have Bacillus, Listeria, and Pseudomonas. These are some of the environmental microbes that we may find in the environment. So we use them in our training and validation set. We also created a lab mix where we would mix these microbes in certain ratios with E. coli cells and also use that for training and validation data sets. Now, after we developed our training and validation data set, we developed the food samples, which are coconut water inoculated with E. coli, spinach water. We also took egg water and inoculated them with E. coli. Now, when we took coconut water, this is a processed juice, so we did not expect it to have any background microflora. With spinach water, since it came from a fresh produced sample, we expected some microflora to be there. And in ag water, we have significant number of microbes in the environment. These are the irrigation water taken from an open pond. And as you can imagine, the pond has large diversity of microflora in it. So they represent samples with increasing levels of difficulty, as you can imagine, from detection perspective. Next slide, please. So these are some of the test sets, just to illustrate some of the samples. So the coconut water sample, the spinach water sample, we see a lot of organic debris from the spinach leaves. When we started the ag water first, we saw both organic debris plus large class of natural microbiota in the background. So we used selective media to suppress them so that we can have a slightly cleaner sample of ag water. And we use that in terms of developing and testing our model. Next slide, please. Using the fast RCNN model, we can predict in coconut water with very high accuracy. Our accuracy in spinach water as well as in the ag water is relatively high. We do have some cases of false negatives, if you were to call them, or false positives, sorry, and that are especially occur at the low end of the ag water case, where we are predicting certain instances which our ground truth data is not indicating in this case. So this is still a work in progress. We are working on developing these models which can use imaging-based inputs. And our ultimate goal is that to develop a method that can be deployed in the field using automated scanners or other systems. And we're looking at different modes of imaging. This data was collected using the fluorescent mode of imaging, but we're also looking at non-fluorescent, non-label-based imaging methods that can allow us to collect those data sets and model these systems. Next slide, please. So coming to the summary of it, machine learning approaches are effective both for process validation and detection of pathogens. Using active learning approaches, we can reduce the number of samples for spectroscopy, and we are also considering using similar approaches for imaging data sets. We believe that the combination of machine learning and point-of-use measurements and some of the edge computing can enable deployment of these analytical approaches in the field. That's where they are needed, actually, because we can do in our lab with many of these methods, but I think one of the challenges that we have to address still is how do we take it to the field where we may have limited resources, both for data collection as well as data analysis. Next slide, please. I'd like to thank all of you for this opportunity to share some of the research that we are doing, but I'll also be glad to take any questions and or discussions that you may have on any of the points that I have presented today.
Okay, thank you, Nitin. That was an excellent talk. Does anybody have any questions for Nitin today?
Hi.
Oh, go ahead.
I'm sorry. I really appreciate your talk. I learned a lot. I'm a microbiologist, so I just had a question about the edible non-living kind of biological compound sensors that I wrote down.
Yes.
I didn't really understand how you could quantitatively see reduction. Is it linked to the amount of compound you use, or I guess I wondered also, is it only put on a certain place? Is it that more for the water sampling where you take a subsample, or like I was picturing an apple or something where you might, how do you sample the whole unit?
No, I think that's a very good question. So I was very brief in that intentionally because I thought many people with data science background may not be that curious about that aspect, but I'll elaborate. So so the way we created these surrogates are that they are basically DNA encapsulated in biopolymer structures. And the way we measure the response is that these DNA molecules, these are long DNA molecules that we are using in this case, because we found that short DNA molecules and the sanitizer interactions are not effective in representing what happens at the microbial level. So these are relatively long, I would say 4,000 base pair length. Again, not very long, but it's relatively longer. So they're not a primer level length, but they are more at the plasmid, short plasmid level lengths in this case. So what we are measuring with FTIR is the oxidative changes that occurs when these oxidizers such as chlorine or the plasma species interact with these DNA molecules. And what our machine learning models are predicting are the relationship between the level of microbial inactivation and the level of oxidative changes that we see in the IR spectroscopy using DNA as a surrogate in this case. Does that answer the first part of your question?
Yeah, I think you're looking for ratios, changes in ratios, or
not necessarily, because so we definitely are looking for certain spectral bands that do change, but it's not truly a ratiometric change in a way that and it's a highly nonlinear change. So by the way, when we think about DNA oxidation, it's one of the fundamental measurement that we actually have struggled in many systems. Like if you think from radiation treatments to cancer chemotherapies, DNA oxidation is one of the ways cells die.
Mm-hmm.
But to make a measurement in a lab, what you have to do is to extract the DNA and maybe use an HPLC, mass spectrometry, or some of the very high-end analytical methods to measure it.
Mm-hmm.
So we are not quantitatively measuring DNA oxidation. We are measuring spectral changes, like the spectral response due to DNA oxidation. Now, that oxidation response is dependent on the dose, but it's not a quantitative response that we can get in terms of classical ratiometric spectroscopy measurements. So that's where I think that's where the machine learning approach has become very helpful, because we have a nonlinear response from our reporter. We have a nonlinear response from our bacterial cells, which are getting inactivated in that simulated environment. And we are correlating those two nonlinear responses using a machine learning model in this case. So that's the strength of the machine learning model, that are able to take two nonlinear responses and correlate them and classify them into different groups of treatments. So that's the first element to it. Now, coming back to your second question, how do you detect it? So it really depends on where you want to detect it. If you want to detect it in wash water, we actually made small produced like samples. Like basically, it's not a true apple. It's a surrogate for an apple that we would put these things on, and you recover that from your wash tank and sample it. If you want to detect just the water, you could take a water sample and incubate with the surrogate directly. If you want to detect on a surface like a biofilm, for example, we made those surrogates to be a biofilm surrogates in those cases. So we actually try to mimic the biopolymer, which is the ECM, the DNA, and some other lipid components in there, and try to mimic the biofilm environment on a macro scale, and use that as a surrogate for a spectral measurement response.
Okay, and there's no need to concentrate your sample.
No, that's the beauty of it. Actually, is that because one of the critical challenge in analytical measurements, for example, in bacteria, if I could directly measure it, would be that I don't have enough sample to detect it. Like,
right.
So that's why we use these surrogates, and we have high enough localized concentration in those surrogate particles that we can actually measure the response without amplification, without enrichment. So it's a point reading at that point.
Oh, it sounds really good.
Yeah.
Thank you. Appreciate it.
Thank you.
So we've got another question from Jeff in the chat. I can go ahead and read that so everyone knows the question. If I can say that word, not my domain. The
Yes.
bacteriophage are quite interesting. Possibly I could benefit from a clarification. Are you interested in fluorescent tagged bacteriophage to help you detect the cell lysis, if not, how to prevent detection of other species of bacteriophage or prevent false positive from mutation of bacteriophage, or is mutation not possible in the time frame of the test or not likely a challenge with this approach?
Thank you, Jeff, for your comment on that question. I like bacteriophage personally as an approach, so I have a bias in this case. So I appreciate your comment there. So I would say with the bacteriophage and the imaging approach that we're using right now, that the data that I showed to you today, we are using a fluorescent approach, but we are not tagging the bacteriophage. So we use a wild type T7 bacteriophage in our E. coli samples. Now, T7 or other E. coli phages can have highly high specificity for bacteriophages, for bacterial targets. So for example, T7 affects about more than 90% of the E. coli, generic E. coli in the environment. Now, when it lyses the bacteria, what we are detecting is we're using a DNA stain that basically stains the bacterial cells. And we're looking for cells which have a certain morphology, and the cells which are lysed have a different morphology. And we're using basically an AI algorithm to find those different morphologies. Like from a practitioner from my side, it's almost the same problem as trying to find one type of car in a mix of different cars. So we have a shape change. Now, it is a hard problem compared to a car problem, because when you think of bacteria, they are on the size of 1 micron by 2 micron. So on a microscope, I got 4 or 5 pixels in length there, somewhere there. So I'm looking for very, very small changes that my algorithm has to detect in this case. Coming to your question on prevention of detection, I think that comes with the specificity. Mutations can be a problem, that there can be bacteria which are, may not be very specific, which may have already mutated to that phage and may not target. So one of the approach that we have not used, but we have always told our funding agencies, is that it is possible that we can use a cocktail of phages. Because again, we don't have to label our phages. So that means that we can use a cocktail of phages directly to lyse the bacterial targets. In a fluorescence spectroscopy approach, I was quite pleased by the specificity of phage in terms of changing the level of spectral response of the E. coli, specifically even in the presence of Spinach homogenate. So I think I'm quite pleasantly surprised by the level of specificity of that chain that the phage induces by lysing the bacteria and reducing the tryptophan-like fluorescence that may occur in that system. Jeff, does that answer your question?
Got the thumbs up thanks in the chat. Very good. Are there any other questions? All right, well, I guess we can conclude here. Thanks so much for your time, Nitin. I know that you've expressed that you're very interested in potentially talking with folks at the lab beyond just our seminar today. So if you'd like to continue the conversation, I'm sure, Nitin, you would appreciate an email from any folks on this call to continue collaborations, right?
And I just, yeah. Definitely, we are very excited about collaboration, and we're working on many interesting fronts in the food systems. For example, we have problems in 3D printing of foods, and we're trying to understand how to make predictive models there. We are working on some problems of developing more sustainable processing technologies using chemistry and physical processes. Again, we're looking at some people who may have interest in more like drug discovery models. We think, again, our data may be a little bit sparse or not that extensive as drug discovery models. So we're looking for collaborators in both those domains, as well as the data that is shared today with spectroscopy and imaging methods.
Great, thanks. All right, well, I guess that's all for today, guys. Next month, we actually have two
Thank you.
seminar speakers coming. I just wanted to do a quick advertisement for those. We have Professor Edelman from MIT, who's going to discuss the Julia language with applications to several domains, including a long list: climate science, material science, and uncertainty quantification. And then following that, we'll have Nusha Amaji from Stanford talking about modeling consumer water demand and implications in the policy space. So a lot of great talks lined up for us next month. So I'll see you all again then. Thanks, everybody.